Hello, I'm Bob Lefkowitz, and I'd like to welcome everyone to day 2 of the inaugural Research Week here at Duke, a week-long celebration of basic translational and clinical research. First, a couple of reminders. Our event today will be recorded. If you have questions during the presentation, please select the Q&A box at the bottom of your screen to share. At the end of the presentation, we'll do our best to try to answer some of those questions. Now it's my great pleasure to welcome all of you here today for the 2020 Robert J. Lefkowitz Distinguished Lecture. When the School of Medicine established this lecture, I was quite honored. I wanted to use the lecture as an opportunity to bring highly distinguished scientists to Duke. I think we've fulfilled that goal through the years, and today is no exception. I'm absolutely thrilled to introduce today's distinguished speaker, Dr. Helen Hobbs. Helen is an investigator of the Howard Hughes Medical Institute and a professor of medicine and molecular genetics at Dallas Southwestern. She received her medical degree at Case Western after graduating from Stanford, and then was a house officer at Columbia University College of Physicians and Surgeons, my very own alma mater. After that, she moved to Dallas Southwestern, where she was chief medical resident, and then a research fellow with Nobel laureates Michael Brown and Joe Goldstein. For those who aspire or who are already physician scientists, she is the quintessential role model. The great respect that I personally hold for her in her clinical role is evidenced by the fact that for many years I have consulted closely with her on many occasions about management of my own problem with hypercholesterolemia and coronary artery disease. But in addition to being a gifted and devoted clinician, Helen's science is dazzling for its originality, for its fundamental insights, and for its clinical translation. Together with her colleague, Jonathan Cohn, she established the Dallas Heart Study, a population-based study of heart and metabolic diseases, which tested the hypothesis that DNA sequence variations with major phenotypic effects can contribute to complex disorders. In the course of these studies, they discovered a number of gene variations which lead to significant alterations in LDL cholesterol and coronary artery disease, as well as others involved in susceptibility to fatty liver disease. These startling discoveries have led to the development of entirely novel classes of therapeutics, such as PCSK9 inhibitors, which have dramatically rewritten the playbook for how to deal with severe hypercholesterolemia. For her scientific work, Helen has been often awarded. She's been elected to honorific societies such as the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. She's received prestigious prizes such as the Breakthrough Award, the Grand Prix Award of the French Institute of France, and the Harrington Prize for Innovation and Discovery. I'm really thrilled to have Helen here to give the Lefkowitz Lecture this year, and proud to introduce her. Helen, we're all looking forward to your remarks.
Oh, thank you so much, Bob, for that really nice introduction. I just got to get my slides on here. There we are. And it's such an honor for me to give this lecture and to have the opportunity to share with you our use of genetics to inform how dietary excess causes two common diseases. As you know and heard, I am a physician scientist, and that means that I have two lives, and one in the laboratory and another in the clinic. The breakdown in my life is not 50-50 as shown in this picture. I spend 90% of my time in the lab and only see a handful of patients every week. There we go. Okay, so this is the picture of the physician scientist. And as I said, I spend 90% of my time in the lab, and and the remainder, I see a few patients a week. But this double life that I lead is made more complex by the fact, as you heard, that I also am a physician to scientists. And among those, yours truly, Bob Lefkowitz. It's even more complex than this, since Bob is not just a scientist, but is also a physician scientist. So I not only answer his questions about lipid levels, but also about pathophysiology of lipid-related disorders. And as you can imagine, he's a tough patient who always keeps me on my toes and always keeps me up to date. When I was a young scientist, one of the very first talks I gave was at the Keystone meeting. Arno Matulski, a prominent human geneticist, introduced me. I didn't know him personally, so imagine my surprise when he introduced me as his granddaughter. I was completely flummoxed. What in the heck was he talking about? Well, he was talking about my scientific pedigree. Arno Matulski trained Joe Goldstein. Joe Goldstein trained me. Therefore, I am Arno's scientific granddaughter. Now, we all know, and you heard, Joe Goldstein has worked hand in hand with Michael Brown for over 45 years, and their work culminated in them sharing the Nobel Prize. And the closeness of their scientific relationship has led to some referring them to twins, fraternal, of course, as I've shown here. Well, these twins have a scientific brother, yours truly. Here are the three of them interviewing each other for an article about physician scientists and the Journal of Clinical Investigation. So what does that mean here? That means I am Dr. Lefkowitz's scientific niece. After today, he may not want to claim me as such. At any rate, today what I'm going to focus on are two highly insoluble lipids, cholesterol and triglyceride. Cholesterol, known well to all of you, planar molecule that is an essential component of all cellular membranes. And in fact, shown here is red cells and with one white cell, but 50% of the cholesterol that circulates in your blood is not in lipoproteins. It's actually in the walls of the red blood cells. Triglyceride is comprised of a molecule of glycerol to which is attached 3 fatty acids. And triglycerides are the major energy source for all vertebrates. And we have specialized cells, of course, in our body that sequester the triglyceride and make it available when it is needed as an energy source. The problem is when lipids are in the wrong place. Cholesterol, as you know, over the years builds up when it's at high levels in the blood, builds up in the walls of the coronary arteries, resulting in coronary atherosclerosis and sometimes causing myocardial infarctions, still a major cause of death in the Western world. And triglycerides, when they're not sequestered in adipose tissue, and in some people, when they gain weight, they deposit triglyceride in their hepatocytes in the liver, and this can lead to the associated inflammation and fibrosis leading to cirrhosis. And so I'm going to give one example of how we've used genetics to tease out the relationships between accumulation of these lipids, in this case in the blood, in this case in hepatocytes, and the sequela disease. So I'm going to start with LDL cholesterol, and I'm going to tell the story that Dr. Lefkowitz referred to in the introduction. This slide shows the relationship between plasma LDL cholesterol levels, shown on the x-axis, and coronary heart disease deaths on the y-axis. This from the United States, from the Mr. Fitz study, and this was a study performed by the great epidemiologist Pito, who wanted to look at the relationship in lower ranges of cholesterol, and to do that had to go to China. And what you can see is basically this data extends the curve. It's not linear, there is curvature here, but a near linear relationship between plasma levels of LDL and coronary heart disease deaths. Now, associated with this difference is plasma cholesterol levels is, of course, a difference in coronary artery disease, with about a 14-fold increase in coronary artery disease in the United States when compared to, for example, rural China. So here's the problem. The problem is, even though this relationship looks so good, is it really due to the LDL cholesterol, or is it due to something else? This is the conundrum that occurs with epidemiology, this Gordian knot, particularly in metabolism, where so many things are interrelated. This could be due to the fact that people are much heavier in the United States than in China. It could be due to the fact that we consume more saturated fat and cholesterol than certainly they do in rural China. How do you figure out what is really responsible for the relationship? And this was something that, this was a question and a problem that persisted for a long period of time, really until genetics entered the scene. And genetics is the answer to teasing these things apart. I want to briefly review how we got into this, and we started with single gene disorders, major Mendelian disorders, major genes, where major genes play a major role in changing plasma lipid levels, in this case, LDL levels. And I just want to remind you about LDL metabolism, that the liver makes a precursor particle, VLDL, the blue here is ApoB, which is triglyceride rich, delivers the triglyceride to the periphery, and to form the cholesterol ester rich particle, LDL, which is cleared predominantly by the liver, and it's cleared via the LDL receptor, the discovery that Brown and Goldstein, my mentors, made. Now, when the cholesterol gets to the liver, that you have to get rid of cholesterol. Every cell in the body can make cholesterol, but no cell in the body can degrade cholesterol. So it either gets made into bile acids, or it gets secreted directly into the bile. And in humans, this is the major way, just by secreting it directly into the bile. And of course, where we really learned the relationship between LDL and coronary artery disease was in the study of familial hypercholesterolemia and the discovery by Brown and Goldstein of the LDL receptor. These young children who have very high plasma levels of cholesterol, no other risk factors, suffer from myocardial infarctions. Also in Dallas, Scott Grundy discovered that there's a sequence variation, most common among Germans, a missense mutation in the part of ApoB that binds to the LDL receptor. It's a phenocopy of familial hypercholesterolemia and also is associated with premature coronary atherosclerosis. And this was where things stood when we started. And what we wanted to figure out is the relationship between LDL and coronary artery disease, is it the LDL cholesterol? Could also be the fact that you have reduced clearance of LDL from the plasma, other possibilities all involving this pathway. And so we went and found there in Sardinia, there's a recessive form of hypercholesterolemia called autosomal recessive hypercholesterolemia. Using linkage, we identified the mutation that's responsible for that, and that's in this gene we called ARH, also called LDL RAP. That's the official name now of this protein because it's an adapter protein. And this brings me back to Dr. Lefkowitz and also Mark Carone here at Duke, because when we found this protein, people say about geneticists, and it's certainly been true for us, you parachute into the genome, you don't know where you're going to be or what you're going to be studying. And I didn't know anything about adapter proteins when I started, but I learned. And how did I learn? I learned by reading about beta-arrestin and work done here at Duke. And it really helped us conceptualize what this protein was doing and test the hypothesis that proved to be true, that this protein actually links the LDL receptor, which sits in these clathrin-coated pits, to the endocytic machinery, having a region that binds the heavy chain of clathrin, another region that binds to beta-2 adaptin, which is a part of another structural protein in the pit. And again, I can't minimize how important the work done here was for us to be able to tease this out. But again, this involved the same pathway. But the next gene we found, which causes a disease called sitosterol, also autosomal recessive, can be due to two ABC half transporters, ATP binding cassette half transporters called ABCG5 and ABCG8, which we show must associate in the endoplasmic reticulum. They go to the apical membrane of either hepatocytes or enterocytes, the only two cell types in which these proteins are expressed. And what they do is they transport sterols into the bile or in the liver, or in the intestine into the gut lumen. So when you eat, this is taking the cholesterol that you absorb and some of it is getting transported back into the gut lumen. So this protects you against the accumulation of neutral sterols. So a completely different pathway that causes hypercholesterolemia and coronary artery disease. So that told us that irrespective of the pathway that's perturbed, very high plasma levels of cholesterol cause coronary artery disease in the absence of other risk factors, that is in children who have no other risk factors. So these single gene mutations, all found in individuals with exceedingly high plasma LDL cholesterol levels having a normal distribution in the population, what this told us was that high LDL is sufficient to cause coronary heart disease. But what we wanted to know is it necessary? Do you have to have it in order to have heart disease? And to do this, we needed to find people who had low plasma levels of LDL cholesterol and were protected from heart disease. Well, those people we were not going to find in our clinics. We were not going to find them at our hospital, Parkland Hospital, our county hospital, the two hospitals in which we are physicians. So we decided we needed to go into the community, and we performed a community-based study, which Dr. Lefkowitz referred to, and that is the Dallas Heart Study. This has been our genetic discovery platform. Wrote this study, designed this study together with Ron Victor, a specialist in hypertension, as well as my scientific partner. It started when we wrote this study, Jonathan Cohen. And I just want to go over the major design features because the two genes I found today depend upon these design features, particularly the first one. We decided that we wanted to have it multi-ethnic. And actually, we oversampled individuals of African descent. Why did we do that? Because they're the most genetically diverse. It's the oldest population, the most genetic diversity. So 51% of the sample is Black, 32% white, and 15% Hispanic. And I'm going to say now that we would never have found the gene that I'm going to tell you about that causes fatty liver disease if we did not have Hispanics in the sample. Because this is a relatively small sample, 3,557, certainly by today's standards. The second premise was that we wanted phenotypic precision. We wanted to measure everything with the very best possible assays available. And the example I'm going to give you today, again, is in fatty liver. We did proton magnetic resonance spectroscopy of the liver in this sample, which led us to a gene. And then I'm geneticist. An individual is nothing without their family. If you find a sequence variation, the very best way to figure out whether it's playing an important role in the trait of interest is to look at the family. So we consented everybody to be recontacted so we could go out and collect the families, which we've done on several occasions. It also allowed us to bring individuals into our medical center and do mechanistic studies. So these are the major design features. But what I want to focus on is the genetic strategy. And our hypothesis, again, was that there are large effect size alleles, Mendelian alleles, that promote health, and they're present in the general population, just as there are ones that promote disease. And that such alleles would provide the most expedient route to function and translation. And if we're going to find these alleles, at this time, everybody is doing GWAS studies looking at common sequence variations. We weren't interested in those. We wanted ones that had major effect sizes for the reasons I will tell you later. And that to find those alleles, we had to do sequencing. So we set up high throughput sequencing. Of course, now that's mundane. At the time, this was Sanger sequencing that we had to do to look for low frequency alleles of large effect size so that we get a handle on function much more rapidly. And as we were doing this, and while our initial studies, we showed that we're riddled with sequence variations. We found mutations in almost everything we sequenced, but the most impactful discovery we made, I'm going to tell you about now. As we were doing this, another group, this was not us, another group in France was looking at patients who had a disease that looked exactly like familial hypercholesterolemia, very high levels of LDL cholesterol, and it was due to missense mutations in a gene called PCSK9. This again shows the power of genetics. This gene had not heretofore had not been implicated in lipid metabolism. And PCSK9 stands for proprotein convertase subtlysin kexin type 9. It's a secreted protein. This is a signal sequence. And then it has these various domains. I'm not going to review. The most important thing to say is that it undergoes autocatalytic cleavage. This happens in the endoplasmic reticulum. And the pro domain sits on top of the catalytic domain. This protein, even though it has the sequences, even though it is capable, it is a protease, an active protease. After it undergoes autocatalytic cleavage, after the pro domain sits on top of the catalytic domain, it has no catalytic activity. And we've shown by Jay Horton in my department here at UT Southwestern is that the catalytic activity is unnecessary for its effects on lipid levels. So this is what is known. These are the sequence variations that were described, but why did they cause a high level of LDL cholesterol? And three groups basically did the same experiment. Again, we were on the sidelines here, just watching what was happening. One of the groups was Jay Horton, my colleague in Dallas. And what they did is they took mice and they used an adenovirus to express PCSK9 in the livers of the mice. PCSK9 goes into the blood. And what they found was that when they express PCSK9, even when they express the wild type form of the protein, the normal protein, as well as if they express the mutant forms of the protein, that the LDL receptor went down dramatically in level in the liver, and the plasma levels of LDL in the mice increased dramatically. The fact that the wild type protein, that is the normal protein, had the same effect as the mutant protein strongly suggested that this was a gain of function mutation. This is so overexpressing the protein naturally degrades the LDL receptor. Overexpressing the protein causes more degradation of the LDL receptor and hypercholesterolemia. Jay Horton told us about these results, so we hypothesized, what will we see in the human population? Well, gain of function mutations, they occur, obviously, they cause disease, we all know that, but they're uncommon. There's so many ways you can kill a protein. There aren't that many ways to get a new function to the protein. And so what we thought was, what would happen if you have loss of function mutations, which are much more common? If you had loss of function mutations, you'd have less PCSK9 getting into the circulation, and you'd have more LDL receptors, and you'd have lower levels of LDL. And based on that hypothesis, we went to our population and we did something extremely simple. We just took the blacks and the whites, the two largest groups in our population, and we sequenced the individuals who had an LDL level less than the 5th percentile. And we hit pay dirt 3 times. First, in the individuals of European descent, 3% had a missense mutation that when we looked at the whole population, lowered LDL, mean LDL levels by 21%. More striking was the fact that 1 out of every 50, 2% of African Americans had one of 2 nonsense mutations that cause a premature stop codon in the protein. And that both these, we showed that this one as well as this one resulted in failure to secrete PCSK9. And this was associated with a 40% reduction in LDL cholesterol. So now we had the molecular handles we needed in order to answer the question that we wanted to answer, and that was, if you're born with a lower LDL cholesterol and you have a lower LDL cholesterol your whole life, as would be the case if you had inherited one of these mutations, would you be protected from heart disease? And so we went to the only biracial study that was available at that time, which was the ERIC study. We collaborated with Eric Boerwinkle, and we did something very simple. The blacks in the studies and the whites in the study, we just stratified them based on their genotype, and shown in yellow are the individuals who have the one of the nonsense mutations or the missense mutation we found in whites. And then we just compared, how did these people do versus these people in terms of 15-year follow-up for cardiovascular events. And I want to draw your attention to the fact that this is a population that has a very high burden of cardiovascular risk factors. Hypertension over half, smoking a third, diabetes 18%. The whites had less burden, but still significant burden. And what we found in this population was that the LDL was reduced 30% in the blacks, in those who had the mutation. And in the whites, it was reduced just 15%. 15% would be like taking half of a Mevacor, our weakest statin. And yet, associated with this was a reduction in the level of cardiac events by 90% in those who had in the blacks who had one of the PCSK9 nonsense mutations, and by 45% in those who had whites who had the missense mutation. So much greater than we see in statin trials. In statin trials, the kind of reductions you would see associated with these reductions would be about 30% and 15%. So we see a greater reduction. And we now know that that greater reduction is because they've had a lower cholesterol their whole life. How do we know that? Because when we look at other sequence variations that alter the plasma levels of LDL cholesterol, that is DNA sequence variations that alter plasma levels of LDL cholesterol, they're all associated with a greater effect size than we see in the statins. And this brings home the point that when it's been referred to as the LDL iceberg effect. So plasma LDL levels are what we see, and when we give a statin, statins are very potent agents that reduce the LDL cholesterol level, and we're very pleased with this effect as physicians. But the problem is, and we know this from the statin trials, that most people in statin trials go on and have a myocardial infarction. Why is that? Because this is not the only LDL there is. There's the cumulative LDL exposure, and this cumulative LDL exposure is still great. You can't turn that around in a day. You can't turn it around. But so why is it different for the PCSK9? The reason why people with PCSK9 mutations have such dramatic protection from heart disease is because they have had a lower LDL cholesterol their entire life. And so they've had a much reduced, they have a much lower cumulative LDL cholesterol exposure. So what you're not seeing, that is what's below the surface, is much less than in a person who's had a higher LDL their entire life. So this work made PCSK9 a therapeutic target. And we also found an individual in the Dallas Heart Study by going out and collecting the families of the people who had the mutations. We found somebody with no PCSK9. She had an LDL of 14. This told us that PCSK9 is playing a major role in raising LDL levels in you and me, in everyone. And I just want to go over, how does PCSK9 work? We perform studies to elucidate this pathway, and I'm just comparing it with the LDL receptor pathway, where LDL binds to the receptor, gets taken up into endosomes, and in the low pH of the endosomes, as you know, the receptor goes back to the cell surface, and the LDL goes to the lysosome, and then the cholesterol is released into the cell. PCSK9, we mapped where PCSK9 binds to the LDL receptor. It binds in this repeat in the LDL receptor called an EGF-like repeat A. When it goes into the lysosome at a lower pH, the affinity of PCSK9 for the receptor actually increases. And this results in the receptor getting degraded along with PCSK9. So causing the degradation of the LDL receptor. So I just want to review what I've said today. So when PCSK9 binds the LDL receptor is degraded, and that results in higher levels of LDL. We all have PCSK9 circulating in our blood. That PCSK9 is raising our LDL. And individuals who have mutations in PCSK9, they have more LDL receptors on their cell surface, so they have lower levels of LDL. So therapeutically, it became clear once the interface between PCSK9 and the LDL receptor was crystallized by Hans Deisenhofer here at UT Southwestern, and it was shown that this is too large for a small molecule to interrupt, that antibodies would be the best way to go. And two companies made antibodies that bind to PCSK9, so it cannot bind to the LDL receptor and degrade the LDL receptor. And those antibodies are very effective. And this is just one of them, anti-PCSK9 antibody in people that are already treated with statins. This is placebo. This is one of the antibodies, and you can see this dramatic lowering of plasma LDL cholesterol levels. And there have been studies that show that it also lowers coronary heart disease. And now 2 such agents are on the market. So this is an example of how a human mutation conferring disease protection has led to new therapeutic agents. This is not the only example, and I'm going to provide you with some other ones that have been developed and are being developed. We've talked about PCSK9. There was a drug that was developed for HIV, because we know that people who have mutations in CCR5 do not get HIV. So this is an anti-CCR5 agent, but it's not used anymore because we're so good. The protease inhibitors are so effective in treating HIV. There is an insomnia agent that is caused by that attacks these receptors. These are the orexin receptors, and an agent has been developed based on the studies of orexin deficiency. And there is a agent now for osteoporosis, new antibody that's on the market, that was developed by individuals who saw that that people who have dense bones have a mutation in sclerostin. So sclerostin is a secreted protein, so they made an antibody. So good examples of how taking advantage of careful phenotyping of humans has led to an identification of loss of function mutations has led to the development of drugs. And in development now, PCSK9, the antibodies have to be given every 2 or 4 weeks. Now there is an RNAi that's been developed by the medicine company, and that you only have to take every 6 months, and it has just finished phase 3, very effective. And ANGPTL3, another protein that we worked on and identified loss of function mutations. Mutations in this protein lower not only triglyceride, but also LDL, but also triglyceride, and ApoC3 lowering triglyceride. And everybody has been intrigued by this disease that was identified that's due to mutations in this sodium transporter, where these children do not have pain. And there's been many groups have tried to target this transporter. It's proved to be very difficult, but it still holds promise in developing a new way to handle pain, which of course we really need. So what I've told you about is one example where human genetics has helped clarify the relationship between cholesterol and a disease. And I want to turn to another disease, which is fatty liver disease, and the role of triglyceride in fatty liver disease. I can say when we started, it was thought that triglyceride was an innocent bystander, that triglyceride, hepatic steatosis, because people who develop fat in their liver can lose weight, and when they gain weight, can lose weight, and the fat goes away and the liver is fine, that this is benign. And in fact, the hepatic steatosis has been referred to as bland steatosis. And so we wanted to set about use genetics to see whether this is indeed the case. Is triglyceride really so bland? So this is what hepatic steatosis looks like. There is triglyceride in lipid droplets in the cytoplasm of the liver cells. It's now the most common liver disease, most common disease seen by hepatologists, and it soon will be the most frequent indication for liver transplantation. And that's due to the fact that hepatitis C now is a curable disease. And so this is a disease that occurs in association with obesity, insulin resistant, and we now know alcohol it has a similar pathogenesis. And we know that because, as I'm going to show you, the genetic underpinning so far has been identical. So these things cause steatosis. In some individuals go on to get steatohepatitis, so-called NASH, and then cirrhosis, and some go and get go on to get hepatocellular carcinoma. So we were interested in the genetics of this disorder. There had been some indication that there's familial clustering of this disorder, and we wanted to know, is there a genetic underpinning to who does and does not get this disease. And so we looked at the prevalence of hepatic steatosis in the Dallas Heart Study using proton magnetic resonance spectroscopy. And that was the largest study that had been done. Before this had been a study of about 20 people, 25 people, and we took it to the population. And what we found was that this is showing the hepatic triglyceride content, the distribution highly skewed with a median of 5.5%. Fully 1/3 of our population had hepatic steatosis. But what was more interesting to us was the fact that the prevalence differed between racial groups, with Hispanics being oversampled and Blacks being having a relative paucity of hepatic steatosis, despite the fact that there were no significant differences in the major risk factors for this disease, which I already went over, which is BMI, obesity, and associated insulin resistance, as well as alcohol intake. So we also, but we found, we did a GWAS study to see if we could find a gene sequence variation that contributes to these differences. And indeed we did. PNPLA3 still the most important and strongest risk factor for hepatic steatosis in the population. PNPLA3 stands for patatin-like phospholipase domain containing protein 3. It's expressed predominantly in adipose tissue and liver. Little was known about it when we found it. It has what's referred to as a patatin-like domain. Patatin is a major protein in the plant kingdom, and it has acyl hydrolase activity. And this is the catalytic dyad. I draw your attention to this serine. This serine is absolutely required for the catalytic activity of this enzyme. And this was the sequence variation that we found, an isoleucine to methionine substitution at position 148. And just here is showing the median hepatic triglyceride level in those who had no copies of this risk allele, those homozygous and heterozygous for the risk allele. So that if you're homozygous, a twofold elevation in median hepatic triglyceride. Also of interest to us was the fact that Blacks, whites, and Hispanics, the allele frequency was mirrored the relative prevalence of the disease in these three populations. And indeed, this single variant explains about 60 to 70% of the difference in the prevalence of this disease in these three different populations. I draw your attention to the fact that among Hispanics, the allele frequency 49%, so 25% of Hispanics are homozygous for the risk allele. Now, importantly, we had to address the same question we did with cholesterol. Is this a primary or a secondary effect? Since both body weight, BMI, HOMA-IR, which is an indicator of insulin resistance or insulin sensitivity, it could be that this confers, people with this are more obese, and that's the reason why they have fatty liver. That was not the case. There was no association with BMI, no association with HOMA-IR or with plasma lipid levels. But that does not mean that these factors don't contribute to the expression of this disease. They have a major effect, and I'm going to show you that here. Here we have stratified the Dallas Heart Study based on their body mass index to lean all the way to massively obese, and then by their genotype, which is the whites again, or the people that are homozygous for the non-risk allele, and the blues homozygous for the risk allele. So if you stay thin, and you have this variant, chances are you are not going to have steatosis. But if amongst the massively obese, almost invariably you're going to have steatosis if you're homozygous for the risk allele. And it's not just steatosis. We had to go to a large population. We went to the Copenhagen Heart Study, and we showed that also elevated liver enzymes associated with NASH and cirrhosis were the effect of this variant was magnified when coupled with obesity and or insulin resistance. And this is showing that a very strong, one of the strongest gene environment effects that has been described. And I should say that another group has shown that this exact same relationship with insulin resistance. So why does insulin resistance cause this? It's because this gene is highly regulated. This is in the mouse. If you feed a mouse an ad lib diet, we set that to 1. We're looking at the messenger RNA. You fast the mouse, there's almost no PNPLA3 expressed. And then when you feed the mouse, you see this dramatic increase, 80-fold increase in PNPLA3, one of the most regulated genes in response to food intake. And we now know that insulin resistance, obesity, of course, are associated with higher insulin levels. And as luck would have it, this gene is regulated by none other than SREBP1C. Why do I say this was surprising? And but maybe perhaps not. This is a gene discovered by a transcription factor discovered by Brown and Goldstein that plays a major role in regulating the enzymes that are necessary for the production of fatty acid and triglyceride. So it also, we showed using ChIP-seq that this is a direct target gene of SREBP1C is PNPLA3. That's what's responsible for this. And so PNPLA3 also, I'm not going to show you, 90% of it is on the lipid droplet. So PNPLA3 is on the droplet as where the triglyceride is. We modeled what does this mutation do? And so this is the catalytic site. This is an isoleucine. This is the isoleucine that is substituted. It sits in a cleft that is hydrophobic. So this is presumably where the lipid binds when it's acted upon by this enzyme. And the methionine is predicted, and this I did with my colleague Nick Grishin in Dallas, to provide steric hindrance to this site. So we would think this is a loss of function mutation. And indeed, when we purified the protein, we expressed it in insect cells and purified the protein, what we found is that it has triglyceride hydrolase activity. By that, I mean these fatty acids, it releases the fatty acids off the glycerol to form glycerol and 3 free fatty acids. And what we found is that the I to M mutation lowers the activity of this triglyceride hydrolase by 80%. So this looked like it was going to be a very simple story. Here you have a protein on the lipid droplets, and there's a loss of function mutation. It's a triglyceride hydrolase. You can't release the triglyceride from the lipid droplet. But guess what? That was not the case. How did we know that? Because knockout mice, 2 groups had made PNPLA3 knockout mice. In their hands and in our hands, you could not get fatty liver in these mice. But we did get a mouse model, and we knocked in the mutation into the mouse PNPLA3 gene. And just like humans, when mice were fed a chow diet, if they were fed a chow diet, then the mice that were homozygous for the risk allele did not have elevated levels of triglycerides when compared to those that are homozygous for the non-risk allele. But when we gave a high sucrose diet, remember a high sucrose diet would turn on insulin. Insulin turns on the expression of this gene. On a high sucrose diet, you see this stepwise increase in hepatic triglyceride level. So very similar to what we see in humans. So then we wanted to know what's going on here. Why are they getting this steatosis associated with this allele? Well, first thing is the variant did not affect the expression of the gene. And this is just showing the messenger RNA levels, showing that when we fed these mice the high sucrose diet, the messenger RNAs were the same. But then we isolated the lipid droplets and looked at the PNPLA3 in the lipid droplets, and that's shown here. These are the wild type. There's so little PNPLA3 you can't even see it with the antibodies that we used, but easy to see in the mice that are homozygous for the risk allele. We also made mice where we knocked in the obligate loss of function mutation. Remember, I told you this is the catalytic serine in the site. We see the exact same thing. And this was very specific. We did not see it for the other lipases that are in the lipid droplet or a resident protein. So a very specific increase in the protein. Why is the protein accumulating on the droplet? That's the most recent question we've addressed and have come up with this answer. What we find is that PNPLA3, the wild type protein, it goes to the lipid droplets made in the cytoplasm, goes to the lipid droplet, undergoes a process where it is tagged for degradation, and the tag is ubiquitin. Ubiquitin is added to the protein, as this is a well-characterized pathway, and then it goes to both the proteasome and to the autophagosome and gets degraded. And what we've shown is that this mutation inhibits the ubiquitination of the protein. It's very poorly ubiquitinated, so it has a very, very long half-life and accumulates on the particle. We think that this accumulation is interfering with triglyceride mobilization. And if this is true, then if we were able to get this PNPLA3, the excess PNPLA3 off the droplet, we should cure this disease. And we've done the experiments. I'm not going to show you the data to show that this is indeed the case. We can use RNAi to stop the production of PNPLA3 in these mice, and they do not get fatty liver disease, because if you don't make the PNPLA3, you don't get fatty liver disease. And we've also used another technology called PROTAC, where we can get rid of the PNPLA3, the mutant PNPLA3, and there's no associated fatty liver disease. And you can imagine now there are multiple companies where are targeting PNPLA3, trying to figure out ways to get it off the droplet or to reduce its expression so that there's no fatty liver disease. So what I can tell you is in multiple studies in many, many populations, it's been shown that the odds ratio of having fatty liver disease, if you're homozygous for the risk allele, is 2 to 3-fold higher. Now, is this related to the fact that it's also been found that these individuals had 3 to 4-fold, and 5 to 12-fold higher risk of having progressive liver disease, these other phases of liver disease? Is that because PNPLA3 is doing something else, or is it conferring some other risks for liver disease, or is it because of the accumulation, the chronic accumulation of triglyceride? The approach we took to answer this question was, again, human genetics. We went back to our population to try to find another gene that causes fatty liver disease by another mechanism. And I'm going to just be very briefly tell you about what we've learned. So we went back, and there is a gene called TM6SF2, nothing really known about it. It's of lower frequency, and it's a missense mutation that is associated with fatty liver disease, associated also with elevated liver enzymes and a lower LDL. It's associated, another group, back-to-back papers in Nature Genetics, another group showed that this variant is associated with lower levels of coronary artery disease. So that dispels this idea that fatty liver disease is causing coronary artery disease or contributing to coronary artery disease. It's not, because in this case, you have coronary artery disease, you have fatty liver disease, and you have protection from coronary artery disease. So it's a polytopic protein. This is a sequence variation. It's localized in the small intestine and liver. And when we did localization studies using immunocytochemistry, it's localized to these 3 compartments. Unlike PNPLA3, it's not regulated by food intake. And when we express this in cells, it increased the turnover of the protein. This was consistent with this being a loss of function mutation. And indeed, when we knocked out the gene in mice, we saw exactly what we see in humans: increased hepatic triglyceride content and decreased plasma lipid levels. And this is on a chow diet. So you didn't have to have any special diet. And I'm going to go to the punchline of what this thing does. I introduced this pathway to you before. You make VLDL, it's triglyceride rich, and it matures into LDL. In this case, you make the same number of particles. There's no problem with the secretory pathway. We've shown that. But they're lipid depleted. They're triglyceride and cholesterol ester depleted. And so they have much smaller particles, which we've shown, and lower levels of LDL. And when we do EM, we can see this is the Golgi. These are the particles here. Compare these to these little tiny particles here. So they have much smaller particles. And so this protein plays a major role in the lipidation of particles. Let me back up here. So what we thought was the best way to get a handle on what this protein does, what it associates with, was to isolate the Golgi, because there's this big difference. And so we tried to do that from mice. We were unsuccessful. So we made the mutation in rats. And rats this is oil red O staining, showing that there's accumulation of triglyceride in the liver as well as in the intestine. And here we could isolate the Golgi, and it's not in the Golgi. I can tell you that it's not in the Golgi. It is in the ER, in the smooth ER, which is a specialized region of the ER for drug metabolism and also for lipid metabolism. And it takes the ApoB containing lipoproteins, which are made here in the ER, shown here, and it basically puts lipid on these particles. So you need to have this to get the lipid onto the particle. And this gets transported to this intermediate compartment, and then it takes it back. And we think that this cycle is what's important for getting the lipid, the particle lipidated enough so that it can be secreted with a sufficient amount of lipid on it. We're testing this hypothesis now. So the take home here is that we found two genes, two different pathways, both cause hepatic steatosis. The first, PNPLA3, lipid droplet protein, disrupts triglyceride mobilization and release of fatty acids. TM6SF2, an ER protein, and it disrupts VLDL triglyceride secretion. And both of these proteins, I've showed you some of the data for PNPLA3. It's true for TM6SF2. They're also associated with these downstream processes. So what we know is that this disease is reversible all the way up to NASH. If people with NASH, if they undergo bariatric surgery, their liver can look normal again. But the problem, we think, is that it's the constant presence of triglyceride in the liver in some is associated with inflammation and fibrosis. So, want to wrap up? So what I've told you is two diseases, and both diseases take decades to occur, except for in unusual circumstances. Coronary artery disease, due to the accumulation of cholesterol in the walls of the arteries, and fatty liver disease, starting with the accumulation of triglyceride and then associated with inflammation and fibrosis. And we know for coronary artery disease, usually we start treating, and we treat in middle age in general with cholesterol lowering agents, and we have many, and we're being more and more liberal about giving those agents to prevent heart disease. We know this is good. I will predict that lipid lowering will also be an excellent way to attack this disease, fatty liver disease. But of course, the best way to attack both of these diseases is diet, a low cholesterol, low fat diet that also provides sufficient calories, but not excess calories, so that there's no associated obesity and insulin resistance, and that cholesterol levels stay low, just as they are when you're born. So again, told you about lipids in the wrong place, these two lipids, and this resulted in the identification by our group of PCSK9 as well as ANGPTL3 as targets for treatment of hypercholesterolemia, and PNPLA3 is still in the works in terms of developing agents to combat this risk factor. And I want to end by sharing with you the people who did this work. I've worked with Jonathan Cohen for over 20 years, side by side, starting with the Dallas Heart Study, and he is my longtime scientific partner. These are the individuals who contributed to the fatty liver work, as well as Julia Kozlatina, an outstanding statistician, and the mice were made by Regeneron. And I want to end by thanking Dr. Lefkowitz for the honor of being able to give this talk today to a place that I've always been very fond of. And I shared with Dr. Lefkowitz before this talk, I have a plate behind me that is I talked at the 75th anniversary of your great institution, and I'm totally honored to be able to have this opportunity to share some of my work with you. Thank you.
Thank you so very much, Helen, for a really outstanding lecture. I mean, just a delightful hour of science. I loved your family tree. I will henceforth be delighted to claim you as my niece. We'll have to look into some of the other ramifications of this, like people like Brian Kobilkin are now first cousins of yours. we can
Yes.
deal with that at another time. I'm going to ask you one quick question. I mean, there's just so many interesting implications, ramifications of your talk. As you know, something I've always been very interested in is how low should we go on LDL cholesterol? I really got a kick out of seeing that early slide that you showed relating LDL levels to incidence of coronary disease in various populations. If I'm not mistaken, you sent me that slide probably 20 years ago or more. If I remember correctly, at Dallas Southwestern, the tradition is that the attending physicians, when they present grand rounds, give a handout of material. And if I'm not mistaken, in my recollection, John Dietschy had given
Absolutely.
a talk on cholesterol, and he showed that. And the interesting,
Right.
okay, so the interesting thing about that is in those days, and you had to go all around the world to get to LDL cholesterols of 70. This was pre-statin, of course.
Right.
Then with statins, we've been able to get down even lower. And now, thanks to you and your work with PCSK9s, there seems to be really no limit to how low we can go. So a couple of questions come up. One, what is, if you will, the physiological level of circulating LDL? What do babies have? And what would you predict based on the affinity of LDL for its receptor? And what about these studies recently, which are taking LDL cholesterol down to 30 and 20, similar to what homozygotes have with the PCSK9 mutations? I mean, surely cholesterol is doing something, and we don't want to go too low, I guess, although, as you point out, there seems to be nothing wrong with your homozygote. So how low should we go? And how low, I guess more to the point, how low can we go?
Okay, so one important point is we're all born with a much lower cholesterol, and we're the same cholesterol in China as in the United States. The increase in cholesterol levels, as you know, cholesterol levels, LDL levels increase as we get older. And that has to do with diet. And there have been multiple studies that are all consistent with that. In terms of how low can you go, there's low cholesterol that is no good for you. And that is low cholesterol in proteins. If you have mutations in proteins that are associated with production of lipoproteins, like ApoB. If you don't have any ApoB, or if you don't have any MTTP, which puts the triglyceride onto the lipoprotein, if you don't have any either of those proteins, you can't transport lipoproteins. Are the ApoB containing lipoproteins are essential for transporting fat soluble vitamins. So those people have real problems with vitamin E deficiency, vitamin A deficiency, vitamin K deficiency. So they have real problems. But in terms of clearance, when you accelerate clearance, so far there have not been associated problems. And what I mentioned was we did find an individual. This individual has a PCSK9 level of 0, and she has an LDL of 14. And she is, she's fertile, she's had children, she has normal intelligence, she doesn't have any problems in her intestine and the brain, the intestine, no problems in her kidneys, all these places where PCSK9 is made, she has no problems to date. And other individuals have been identified. There are about a handful of individuals. So it's difficult, but nobody has seen it. And in the trials so far, there has not been any associated problems with LDLs that we used to say we predicted would be problematic, and used to be very careful about how low we took the LDL. So right now we don't know. I think people are not as comfortable with LDLs lower than 25 milligrams per deciliter, but we don't panic about it. Nobody panics about it because it doesn't seem to be problematic.
Got it. Thank you very much. Now, we have a whole raft of questions, not surprisingly, that come in. We don't have time for them, alas, but so I'll just put forth one, and I'll take the moderator's prerogative of choosing the one. Let me read you a comment and a question from your old colleague, Chris Newgard. So Chris says, "Hi, Helen, great talk. We recently found changes in phosphorylation of PNPLA3 in phosphoproteomic data sets. Besides the interesting effects of specific mutations in the gene, has your group encountered or studied any effects of post-translational modification on PNPLA3 protein function?
Okay. Yes, that phosphorylation was originally described by somebody here at UT Southwestern and has been working with Nabil Sadeh to characterize that. And a paper just came out on the phosphorylation of PCSK9 and exactly what it means in terms of how it impacts on lipid metabolism is not clear yet. It also we studied the tyrosine sulfation of PCSK9, and that occurs, but we did not find any effect of that post-translational modification. There is not a crisp relationship. There is a relationship between circulating PCSK9 levels and, heart disease. But there, I think you raise a really important and interesting question. There could be other factors that are impacting on this relationship, and post-translational modifications are one of them. There's so many questions about PCSK9 that remain to be answered. How it circulates in the blood, how it gets degraded, what are the factors that impact upon its half-life? These are all issues that need to be explored.
Well, I think thank you very much for that, Helen. We're just over time now, so we're going to wrap it up with apologies to those whose questions we couldn't get to. So let me conclude by just thanking Helen for an absolutely fantastic lecture, for showing us what I think is basically a paradigm for how to combine fundamental biomedical research with clinical translation, really the very best of bench to bedside research. Helen, thank you again. We're in your debt.
Oh, you're too nice. Thank you. Thank you, everyone, for your kind attention. I appreciate it.
Okay, bye-bye.